Good evening, everyone. My name is Chrissi Kelly. I'm the founder of AbScent, as you know by now. Welcome to the 21st in our series of Tuesday evening webinars. Hard to believe it's been that many already. I think, Simon, you've been with me now. This is the fourth time. And for Jane, this is the third time you will be with us talking about various aspects of life with smell loss. And as always, or not always, sometimes we have Aidan Kirkwood.
Part-timer.
He's a part-timer now that he's doing his PhD at Nottingham. Aidan is manning the Q&A bar tonight. So before we start, let me just tell you a little bit about how that works. If you've got questions as we go along, please put them in the Q&A bar. In the chat bar, I have put a link to the new AbScent network. Now, as many of you know, we have a huge following on Facebook. This is becoming a little bit unmanageable. So we have started something new called the AbScent network. It's a little bit less noisy. And we also have the facility there to offer courses. So right now we are 2 weeks into our smell training masterclass. And next month we will be doing a masterclass on Parosmia. So if any of those things interest you, do please join the AbScent network. You'll find the link in the chat bar and it is free to join. So do please check out that link. Right, and by the way, if you are interested in the smell training masterclass, it is already underway, but we will be running it again. So if you're interested in that, just join the network. Right, so without further ado, let me introduce my guests this evening. I met Jane Parker in March of 2019 when I spoke at a conference for food scientists. And I gave a talk about Parosmia, and that was the evening that I met Jane. And we have been thinking about the problem of, or trying to understand what it is that makes everything taste and smell so disgusting when you have Parosmia. Working with me on that is ENT Simon Gane from the Royal National Throat, Nose and Ear. Simon, I know that hospital has a new name, but I think, do we need to learn that?
They've changed it around to ENT, Royal National ENT and Eastman Dental, which is sad for somebody who's been there 20 years, but
okay,
we've got to move with the times.
Excellent. Okay. And Simon is a trustee of AbScent, and he answers the many, many questions I have every day. So I'm sure it's a bit like fielding questions from a 5-year-old. But anyway, thank you, Simon, as always, for being such a good person about answering my questions. Okay, so I'd like to say one more thing about this research, and this is a very exciting evening for us because this is the first time that we're actually talking about the kind of nuts and bolts of what we found in our research. But the interesting thing about this is that so much of what we have used in our research has come from the patients, people in our Facebook group, and it really does feel like we have brought together in a collaborative way input from the patients. We've created this research and we want to bring it back to all the people who've contributed to it. So in that respect, it's a wonderful model, and I'm so grateful for all the people who have taken time to comment, especially in the Parosmia Facebook group. So anyway, without further ado, I want to hand it over to Jane. So Jane, tell us the story of Parosmia.
Well, I'm quite excited because we've just put out a paper that contains a lot of what we've been doing over the last 18 months. And I thought I would go through some of it, actually. I've got some graphs, especially for the people that have contributed either on Facebook, on polls, on surveys, or especially the people that have come to Reading. I don't know if there's anybody out there that actually came to Reading, but it might also give you an insight to the sort of things we do if you were to come in the future, because there's by no means have we stopped. In fact, we've got more questions than we started with, which is always the way it goes in research. And so we will be recruiting more people. So I do have a few slides which I can share with you. And let's go from there. This is a word cloud that we got out of, I think this was the results from the survey, one of the surveys that we put out. And that's just telling us which are the foods that cause you as Prosmic individuals, what foods actually give you those distortions. And obviously the bigger the letter, the more responses I got. So coffee came up top, as it often does, whether we ask on polls on Facebook or we do surveys or whatever we do, coffee is often always there in the top 3. And then we've got meat and onions, always there in the top 3. Perfume's quite high on that one. Chocolate comes in there in the top few, often associated with coffee. And then you'll see other things. And there's an awful lot of things in there. So, you know, although we've got a key group of offenders, we've got an awful lot of other things like herbs and pastry and detergent and mango and all those other ones that I can't read. So our job was really to find out what it was about these foods that were giving these distortions. And since coffee was up there on the top, and many people drink coffee and we know quite a lot about coffee, we decided to use coffee. It wasn't that we wanted to analyze coffee, but we wanted to use coffee as a source of lots of molecules that we thought might be causing parosmia. So how did we do it? And I think you'll recognize this lovely volunteer here who was one of the first people to go through our
handsome gentleman.
Yep, go through one of our days at Reading. And what we do is the machine just on the right of Aidan actually separates out all the molecules that are in coffee, and they come out almost one by one at the nose piece here. So Aidan's just got his nose just gently resting on a tiny little nose piece there. And this is what our volunteers did for us. So they came in, sat down there and told us what they were smelling over about 20 minutes. So could you smell anything? Could you describe what you smelt? How intense was it? And was it one of those smells that just reminded you of parosmia? Was it one of those sort of really distorted things? And it was quite hard for people to describe what these smells were, especially the sort of parosmic smells, because as you know, many of you, it's just impossible to describe them because they're things that you can't relate to. They're things that you haven't smelt before. So how can you describe them if they're so new? But anyway, we did that. And we had 15 people who came before COVID, before we knew anything about COVID, because this is it's not a new problem. We know that you can get parosmia with other infections such as flu, common cold, sinus infections, upper respiratory tract. And then COVID came along, so we had to stop for a little bit, but it became a little bit easier to find volunteers after that. And we had another 15 come over the summer. And we also had 15 what I call non-parosmics, because they were people that just weren't parosmic. Okay. And the other thing we did with everybody that came through was to assess their ability to smell things. So what was their olfactory function like? And we used a set of sniffing sticks, a bit like felt tip pens. And I'm sure they've been waved around on a webinar before. And it looks at 3 things. It looks at threshold, which is how far can you dilute smells until you lose them. We looked at discrimination, which, you know, how well could you tell 2 smells apart? And identification, could you identify them? And you add all these up and you get a number out of 48. So we had 2 things. We could see how you performed on the GCO, and we could see if that correlated with your olfactory function. So the first thing we looked at was just your olfactory score. So what was your score on the sniffing sticks? And this group here is the group of non-parosmics. And so you can see their average score is about 38, which is quite high because a lot of people were really quite interested in smell. And we got a lot of people that were pretty good. And then we've got the pre-COVID people here and the post-COVID people here. And their average was about 27 or something. So confirming what we knew, really, what you told us, that you couldn't smell things, or you could smell much less than you could before. But the really interesting thing was to look and see how that correlated with how many aromas you picked up on the GCO. So how many times did you pick an aroma? And here we've got a nice graph that shows you the score, your olfactory function, and then up here we've got how many aromas you smelt, and there's a nice line there. And if I tell you what the colours are all about, the yellow people are the experts that are in my group. They've done a lot of GCO, they're pretty well trained, they know what they're looking for, and one of these is Aden. I don't know which one, but one of those yellow dots is certainly Aden. And then we've got the blue ones, which were the non-Prosmics. So a few of those up there, a few black ones who are non-Prosmics, but they had quite a low olfactory function, very similar sort of olfactory function to many of the people with Parosmia, which are the red and the green. And oops, I've lost my mouse, red and green. So the red is pre-COVID and the green is post-COVID. There's no difference between the two. The only difference, which is quite interesting, was age. And the people that were post-COVID tended to be 10 years younger, at least 10 years younger, almost 20 years younger than those that had come to us before. But, you know, this is quite a nice line that tells you if you've got poor olfaction, a low olfactory score, you don't smell very much on the GCO. And that makes a huge amount of sense. We've got a straight line pretty much, and we can correlate those two. But the next thing we correlated was how many of those molecules that you smelt were actually giving you that parosmic sensation. And this is the graph that we get, and there's really no line that goes through that. There is sort of a line, but it's pretty random. And what's so interesting is the fact that it doesn't matter what your olfactory function is, you can still be parosmic or not. So we've got people along at this end who have got a high olfactory score. They can smell all sorts of things, but these people have only got a couple of parosmic odors, parosmic triggers if you want, whereas people with the same olfactory function up here, oh, I keep losing my mouse, up there, they had 10 or 12 different molecules that they picked up as giving you that nasty parosmia smell. And then at the other end, if we go sort of mid-range, again, pretty low score. Some people only picked up a couple and some people picked up 10. So the number of times you perceive this parosmic trigger or these parosmic triggers hasn't really got very much to do with the fact that you've lost or you've got a lower sense of smell. And these two aren't correlated, and that's quite fascinating because it means that for us there's a couple of different things going on. Yes, after COVID or after your infection, your olfactory function went right down. Many people lost their sense of smell, and you know that you're growing it back, and at the moment you're still in the process of it coming back. But the parosmia is different. It doesn't, we had some people who really weren't reporting any loss of sense of smell, telling us that they could find, you know, they had parosmia. So the mechanisms for the parosmic bit, the bit that gives you that distortion, are different. And the really important thing that we found, I think, is the fact that there were specific molecules that would give you this sensation. And we could split them into groups. And I think there's been a lot of comments on Facebook about sulfur compounds, and indeed, that's where we started. We thought sulfur compounds might be involved because they're so potent. And we had a group of compounds, it doesn't matter what these codes mean, but we grouped them and we found a group of sulfur compounds that gave you that sensation, and they were related. We found a group of nitrogen compounds, which gave you usually the same sensation. It was usually the same smell, but some people could tell the difference. There's another group of nitrogen compounds down here, which are very similar to the ones above. They just happen to have separated. And then another group of sulfur compounds down here, which are different to the ones at the top. So we've got, we found that different structures, or we found that groups of compounds with the same sort of structure are responsible. And that is such a fascinating finding for us. And we tried to link this to odoreceptors, and we can't yet link this to specific odoreceptors, partly because we don't know, or we need to know about the pairings between odor receptors and compounds. So there's still a lot of work that needs to be done. But the ones that we do know about were not giving us any information. But we've got a gut feeling that there must be something there, because the structure of these molecules is what seems to be giving them these prosmic qualities. So then we started analysing other things. So could we find them in, what was the problem in chocolate? What was the problem in meat? What was the problem in French fries? And to our surprise, many of the same molecules kept cropping up. So that explains why many of you, when asked to describe meat or coffee, you say, well, it's the same horrible prosmic smell, it's smoky, it's chemical, it's the same smell, because the same compounds are appearing. And we had one volunteer who smelt the same 10 compounds in coffee that she smelt in chocolate. And thinking about the food chemistry point of view, they're both roasted beans, and that doesn't surprise us. They've got very similar compounds in common, but it explains why cocoa, coffee and chocolate smell the same, because all that this person could detect, it was the same group of compounds. And we went on to peppers, and they're a little bit different, and watermelon. Watermelon and cucumber were different again. And we've done bananas, onions, garlic, toothpaste. That was an interesting one. Peanut butter, eggs, toast, cucumber, cola, wine, Clinell wipes, you name it. We've got quite a long list. But the interesting one, and the one that you will all appreciate me talking about, is this one. Because we also know from comments on social media, from surveys, from polls, from questions on Facebook, that this little fellow doesn't seem to cause as much trouble as he used to before. And many people are saying, well, I can't smell anything. I can't smell my own bathroom odours, my own poo. I can't smell it. What's going on? Other people say, well, it just smells the same as coffee. And other people say, well, it smells like distorted coffee, or it smells Parosmia. So we were very fortunate in that we had a volunteer who was willing to sit in front of the GCO and smell a sample of poo. We do quite a lot of that in our department. we work on gut models, and we're always, there's a ready supply of material there for us. So that wasn't a problem, and wasn't anything terribly of any interest really in our department. It's part of the routine. So we put one poor person on the end, and I was the normosmic, because we obviously don't ask volunteers to smell these samples, because you'd never come back, you'd never forgive us. So we had a couple of researchers, and this is what we found. And I've got some names of compounds here because it was just easier to leave something rather than nothing, but ignore the names. So as we were going through this chromatogram, the Parosmic person was picking up a number of smells. And one of them, A, was a roasty, toasty note, and the star means that it was a trigger for Parosmia experience. So it generated that nasty Parosmia smell. And then there was this meaty one, which was normal. And then there was a weedy one, which was Parosmic, and a jammy one, which was normal, and this one burnt nutty, which is very common in coffee, which was Prosmic. And all these are compounds from coffee. So essentially, at the beginning of the run, that's me on the right-hand side there, I picked them all up. Obviously, none of them were triggers. But at the beginning, what these people, or what we were smelling, was coffee. And then it became more interesting, because as you go on, the molecules get a little bit heavier. They're separated almost on weight, really. So they got a bit heavier. And we started to get the compounds with a very typical fecal odour. Now notice the Prosmic individual couldn't smell any of them. F, G, H, and I, they're all a bit fecally, farmyard, manure. And this one at the bottom is a sulfur compound. Nothing. Boy, I was in tears when I did it, because I could smell those compounds so strongly, and it was just, the difference was incredible. And it became clear that the people that are, or most of you that are not smelling poo, it's because you can't smell any of the key compounds. What you are smelling is these things. Some of them are triggers, some of them aren't. So there'll be some people that smell nothing, that makes sense. There'll be some people that only pick up these ones that have got asterisks. So you're the people that smell poo as parosmic. And there'll be some people that smell these ones without asterisks. So you might get a sweet, biscuity, jammy or meaty note, probably depending on what you've eaten, if I can be quite blunt. So that explains why poo is not as bad as normal, because you're just not picking up those aroma compounds. Now, why you're not, I've no idea. I have a little diagram next, which gives you a little bit of, maybe it's one we could talk about.
Jane, why don't we, we'll let Simon talk about that maybe, or Simon, would you like to chime in at this point? Ooh, and he's on mute.
Yes, I'm on mute, but I'm now here. Yeah, I can talk about this for hours. So I think this is so. I mean, I love the fact that Jane, you've managed to explain exactly what we found without getting nearly as excited as I did about finding these things, because
excited.
I think it's fascinating to we now know that there's a molecular basis for this horrible thing that parosmics are suffering from. That there's and once we understand the mechanisms, then we can start to do something about it. But the fact that there's this repeatable trigger across lots of parosmic people, that is a molecular identifier, common molecules between them, means that it must be associated with the receptors. And that really narrows down what's going on here to explain this. It doesn't explain everything, of course, but I think it's a sort of a great step forward. So to understand why I think this is so exciting, you need to understand what's going on with the sense of smell and how the nose is understanding the odors. And that's what Jane's diagram is showing here. Essentially, you have a suite of olfactory sensory receptors on olfactory sensory neurons hanging into the nose, each one of them picking to express just one of 350 to 400 different olfactory receptors. So each receptor, they could have any one of 350, they pick one, and that's the only one they express. And they just make lots of copies all over the surface of the nerve, and it's hanging in the air, as it were, waiting for the smells to come through. And the clever thing is that those nerves also know, if they have, say, the green receptor, which you can see there, they know that they have to grow, they have to pass their little axons, the wiring, up to a part of the brain called the olfactory bulb. And within that olfactory bulb, they have to grow to one of two glomeruli, which are structures in the bulb which have joins to the nerves that go off to the rest of the brain. But though each glomerulus has only basically the sensory nerves expressing a certain receptor growing to it. So in other words, the nerves from that glomerulus to the rest of the brain say, I saw this signal, therefore it was from this glomerulus, therefore it was triggered by that olfactory receptor. So it's a way of mapping this atomic level detection event to a sort of multicellular structure within the brain, and that's what's so clever about it. And it's the pattern of activation of those glomeruli, i.e. the pattern of activation of the receptors, that makes a certain smell the way it is to us. So our brain is recognizing that pattern of recognition. So when we're smelling the coffee, all those compounds that Jane was talking about, the lots of different compounds, they stimulate lots of different receptors. One molecule can stimulate multiple receptors can detect one molecule. It's quite messy, but that pattern of activation, when you take a sniff in the morning, that is what you recognize as coffee. And so, so this now says to us, well, this is why we're starting to see a problem with the loss and regeneration of parosmic smell or hyposmic smell. When you can imagine you lose certain parts of that pattern, you're going to see a new pattern emerging, which you don't recognize as coffee. You smell it, but perhaps only half of the glomeruli are activated. Well, that might be why you're aware that it's not quite the same smell. It doesn't really explain why it's parosmic, and that's really the unexplained part of this research. We have some theories which we'll probably talk about, but we're getting closer, and that's why this is so important.
Simon, could I just clarify for a moment? The terminology is sometimes confusing. So we all know that parosmia is a qualitative disorder. It means that, you know, when a person has it, I mean, anybody in the audience who has it doesn't need to have it explained to them. But what I'd like to clarify right now is when Jane talks about a parosmic trigger or a smell that is parosmic, I think what we need to clarify is that those are smells that are particularly disgusting. So the smell that makes you turn away and want to gag. And of course, that's on a spectrum. So some things will be just odd for people, going right up to the point where being confronted by that is enough to make you want to vomit. So, you know, we haven't really quite yet worked out exactly the right way to describe all these things. But for anyone in the audience who is thinking, well, what's a parosmic smell? It means the kind of smell that provokes disgust. And I think that to talk about disgust in this context is very important, because there is something about the disgust response that I think might lead us eventually to understand more about what parosmia is. So sorry about that interruption.
Yeah, absolutely. In fact, I was thinking we need to define our terms, I suppose. So for me, parosmia has four qualities. Medics love counting things. We love having lists, and we love having numbering things like Buddhists. So parosmia is triggered, and we knew this starting even before Jane started this research. We knew that it was a response to an environmental stimulus. It is relatively short-lived. In other words, it tends to have, not always, and we can talk about the sort of the overlap between smell lock and phantosmia and parosmia, but it tends to be relatively short-lived. In other words, the same kind of a response as you would see in a normal response to a smell. It is altered, and then the key thing that I think you brought up very well, Chrissi, the key thing is this disgust or this unpleasantness. And it's important. I mean, Jane glossed over this. I think note that none of these molecules on their own, to somebody who is normosmic, who doesn't have suffer with parosmia, none of them smell bad on their own. It's not just that you're picking up one molecule at the expense of all the others. It's not just that you've only regenerated certain receptors. There's something else going on, and there's something else that we still don't understand why that should necessarily be so disgusting to us. Now, disgust is a protective mechanism. It's a way of programming us to avoid unpleasant experiences, and there's a lot of research about it. We don't know why that should be, that these smells always trigger disgust, but it is almost universal for parosmics.
Shall I take the screen down?
So Jane, carry on now. Sorry about kind of horning in there on your
No,
your
that
slide
was really
there.
what I was hoping you would do, because I'm at the end, really.
Oh,
the
okay.
slide that I've got is an important slide, because on every time I present this, I do acknowledge all my volunteers. And that's not just the volunteers that have been to Reading, it's the volunteers that have participated in any number of surveys, polls, questions, etc. that we've put out there. And some researchers, well, not just some researchers, but our own Aidan and others who have given up their time for free, really, to come and help us out in the lab. So that's an important slide, actually. But other than that, that's all I have. So I'll stop sharing and we can go back to normal.
Oh, well, thank you very much for that, Jane. I mean, I think we're only beginning to scratch the surface. And I'd like to go back to Simon now. And Simon, imagine with clinical practice, how do you see what we have learned from this research? How do you see that as kind of affecting the way you might counsel patients or talk to patients about their parosmia?
So the embarrassing thing about this is this. I mean, it's obviously an elegant set of experiments, but if I and my colleagues had actually listened to our patients, I mean, we kind of knew this a bit. Like coffee was always a trigger for parosmia, but actually, if you listen to the patient, and this is probably my number one takeaway, listening to your patients as a medic tells you so much more than you think about a condition, especially one as ill understood as smell conditions. But we could have said, we know that there are triggers there, and over and over again. And although I knew it, I didn't think about what the underlying mechanism would be until we started talking about this. That actually, and I didn't know, for instance, that coffee contained these very powerful, and by powerful I mean you don't need much of those molecules to stimulate a response. So they're what we call low threshold odorants. Before talking to Jane, who understood a lot more about that than I probably ever will. But we could have worked out at least some of this by listening to the patients and started a taxonomy of parosmia if we just taken a bit more time to listen to things. And that's where I think AbScent comes in really well. It's a place. One of our earlier papers is exactly looking at this. Is looking at the what patients have to say about their own condition. That can provide us with a lot of insight. So number one, we're not that good. We should have listened to the patients before. Number two, this is really powerful in terms of helping people right now, because I can tell people, yes, this is what to expect. These are the kind of things that you might expect to be triggers. These are the things you might want to avoid. I mean, so some people with parosmia are, and I'm sure I don't have to tell you this, severely compromised by their parosmia, to the point where they can't eat food. They're existing on protein drinks and things like that. And to be able to say, well, if you're eating this, and we've sort of constructed safe food groups, but to be able to understand why those are safe and what to expect would be just a very simple, basic set of guidelines for patients right now. The second thing to say is we talked about what we euphemistically called bathroom odors, and not being, and this is actually quite distressing for a lot of people. Body odor being it's a significant concern for a lot of people who've lost their sense of smell. Not necessarily if they just have parosmia with it, but in general, Jane has shown that they're going to lose these kind of what we think of the characteristic fecal odors, indole, skatole, and to be aware of this and to say, oh, that's normal. You might be concerned about it. It's very unlikely that those body odors are bad, to be and to be upfront about those concerns being, it just allows you to address them and become less of a problem. And then finally, in terms of the treatment, well, if we understand what's going on here, if we understand that there's something about the regeneration of these particular neurons, because what we didn't talk about is the way that parosmia presents. And it seems to be a feature of recovery rather than initial injury. And so our current theory is that these olfactants, these molecules, represent a small privileged set of olfactory receptor neurons that for some reason regenerate early. But because they regenerate without the surrounding neurons, so that pattern that I was talking about in the olfactory bulb is missing very important pieces, or because they don't have the other neurons regenerating, so they cross wire within the bulb. They start activating glomeruli that they shouldn't. That becomes this disgust evoking signal, and that's going to be really sort of the next thing that we have to try and work out, because if it's because these neurons are lonely, well, we can help with that. We have ways of reducing the threshold of other neurons that happen to be there. So we allow them to stimulate easier. And maybe, you know, the smells to then get lost in a crowd and the whole thing becomes less parosmic.
Wait, Simon, can you unpack that a little bit more?
So
how would that be achieved?
Well, I mean, we know there are medications, drops that basically lower the activation threshold of these sensory neurons. And so you could think that if the problem is that these signals are too salient, in other words, the powerful odors override all the other ones and crosswire, and that's what's giving you the prosmic signal. Well, if you encouraged all the other ones to be a little bit more active, you might drown them out and normalize that smell. And so that may be a way that you could improve parosmia. And we know that there's some indications that there's some things we could do towards that. So in terms of therapeutics, that's
yeah, we had a question here saying years ago we found sodium valproate. It helped 1 or 2 parosmic. Might be worth trying again. I think you've said that before. Different sort of GABA-based inhibitors are shown promise. So yeah.
I think I know who that was who said that.
Someone who knows their stuff, I think. Chrissi, we want to move over to questions just now. We've got quite a few in here.
Yeah, let's quickly go through a couple of questions. I'd like to keep the questions, if we may this evening, more about the mechanisms of parosmia rather than questions that have to do with any individual's personal experience of that. We will have other webinars on that subject, but let's try and keep the questions to the mechanisms.
Sure. So we've got a question here from Marianne, who says, do you have any ideas about the mechanisms for parosmia and phantosmia when there is no trigger? I find it interesting that my phantosmia smell of the things that the parosmia person on the slide could not smell, i.e. the fecal smells. Is this typical for phantosmia? Would you like me to read that again? Or?
No. I can answer that. So from my point of view, and there may be other definitions. From my point of view, phantosmia is not a triggered perception. So to me, phantosmia, and this is very difficult I will grant, it's very difficult to separate these out. To me, parosmia is a triggered perception. It may not be triggered by the triggers that we've been talking about, because of course you can have other things that might trigger the same response. But phantosmia is to my mind a hallucination of smell, a smell that isn't there and is not triggered and lingers for longer. The problem is that, like Marianne said, body odors, for instance, might be with you all the time. We habituate to them normally, but who knows that your phantosmia is actually not a parosmia triggered by body odor with poor habituation. So yeah, I think the diagnosis is more parosmic, and Jane, I think would agree with me on this. It was interesting that not everybody reacted to the same triggers. I don't think you showed that necessarily, Jane, in your slides. I know you cut them down, but some people would only react to the sulfur or only react to the others, to the thiols. So that was quite interesting. There are probably several different classes of parosmias, and some people don't have any of those. I think, Jane, you could talk more about that. On mute.
We've moved on to other things now, and we do know a bit more about the things that are similar to coffee, which are cocoa and meat, but we're looking at other things, and the things that we find in cucumber and watermelon are yet another set of parosmic compounds. And if we're thinking about fragrances, I think it's different again. So we've found a subset in coffee that happen to be troublesome, but there are other sets as well. And some people are fine with coffee, but have problems with household products. So that makes it even more interesting and even more complicated in that different people have different experiences and find different groups of compounds troublesome. It is, I mean, the graph of the clusters I showed you, that was based on the 15 people we've seen, and that did cluster. So there was a cluster of people that tended to, that's what those clusters mean, really. There was a cluster of people that smelt the top sulfur compounds, another cluster that smelt the nitrogen compounds more strongly, and another groups that there was a second set of sulfur compounds. So there's some commonality, but there's also a lot of variation as well.
Mm-hmm.
There's a very interesting question from Catherine Bent,
Mm-hmm.
who says, does this mean that it is one's brain that has been affected, and now it is the brain that is giving the wrong signals about what coffee, etc., smells like? Or is it that the nose receptors are giving the brain the wrong information, hence the brain sends back the wrong information? So what do you have to say about that, Simon or Jane?
After you.
Well, I was going to say after you. I think it's both. So we know from Parosmia research previously that Parosmic smells seem to cause different patterns of activation when you look with an fMRI in the brain. The bits, in areas that we can see these patterns of activation represent smell. So I was saying earlier, the activation of the glomeruli, the pattern of activation of glomeruli in the bulb represents a smell to the brain. But you can track that into certain parts of the brain to look at different areas that activate when exposed to certain smells. And you can, and although these vary between people, they're reproducible in one person. So for instance, a rose will give you the same activation in the same area. But you can see in Parosmic people that there is altered activation in those areas. Personally, I think that the upstream parts of the brain, so everything from the glomerulus, from the bulb up, remains roughly the same. But because the bits that it's wired up to are now miswired below that, the patterns that it's seeing are not the patterns that it recognizes. So, in other words, you get in a normal smell, you get pattern activation in the bulb that switches on certain nerves. They send their signals back to the brain. Those are then the brain's thought, if you will, of what a rose smells like. And that hooks into all sorts of things like the emotional valence of smell. That time I got the roses for Valentine's Day, red color, love, or memories, all of those things. That pattern is what that means for the brain. Now, if you can imagine, you mess around with the wiring down below, and when you get the smell of a rose, it triggers ginger, coffee, and bits of something else, to the point where your brain it's unregulated. There's a lot of feedback, and it's perceived as unpleasant. That pattern will not then go back to fit rose anymore. And that's what will be interpreted as not being the same as rose. And a lot of our participants talk about new coffee instead of old coffee, or new whatever, because they'll recognize the new pattern of activation, but it won't link up in the same way to the rest of the brain to give them the emotional other relevant information. What do you think, Gane?
Right, shall we go on to another question then?
Sure. Well, I've got this question up here. We asked here, have you studied the components in urine that might trigger prosmia? Yeah.
No, we haven't.
We're on starting it, yeah.
We thought we were pushing our luck getting somebody to smell poo, but I expect it's the same compounds, because it's the same compounds that are stable as they go through the body. And I very much think that many of the same compounds will come out. Yeah.
You think that could be to do with food as well in that sense?
Yeah, probably what you've eaten, I mean, coffee, you can you often smell that.
Mm-hmm.
So
worthwhile asking them diet and things like that. Yeah.
There was a question that came up, I think it's been asked a couple of times, about the importance of smell training when you've got parosmia. That was one that I'd like to cover now. And the second one, which does come up quite a bit in the Facebook groups and in the network, which is, what about people who have had COVID-19 related smell loss who don't have parosmia? What does that tell us? And are we seeing two different groups? I mean, I know that we heard here in one of these webinars from Thomas Hummel that parosmia is considered to be a good sign because recovery is underway. But what does that mean for people who have no parosmia at all? Simon.
Jane's turn, isn't it?
Pardon? Jane's turn.
Yes, Thomas's work has showed that if you look for predictors of recovery, this is pre-COVID. Parosmia was a good one. Female, young, post-viral. Those are the people who had good recovery. And it's unsurprising that parosmia should predict good recovery, because we now know that this shows that there's regeneration, that those nerves have come back and are active. And if they come back, then the other ones presumably can be helped to come back. I keep meaning to mention the role of smell training. I don't think we should forget about that in terms of, there's some questions about stopping or slowing it down or helping recovery. And I think that if smell training does increase your overall neuronal regeneration and the diversity of those neurons, then with this current findings, I think that there's good reason to expect that would reduce the overall burden of parosmia. Again, the little nerve buddies are going to regenerate alongside these more powerful, more easily triggered nerves, and reduce the noise that they are introducing to the system. What was the other question about separate prosmic non-recovery?
People who don't have parosmia. What's the story behind that?
Well, I think so. It's difficult. I mean, this is all just guessing at the moment. We unfortunately are not allowed to just go and cut a couple of hundred noses out of people who are losing their sense of smell and look at how the nerves are behaving. That's frowned on nowadays. So we just have to guess. But you can imagine that for some reason these selected nerves aren't regenerating in the same way, or they're not miswiring. Then maybe a sort of smaller insults. So I'm pretty certain that COVID-19 anosmia has a couple of etiologies, one of which is just obstruction of the olfactory cleft. So just, putting a peg over the nose. And that's why so many people lose and then recover their sense of smell so quickly. And followed by nerve destruction, probably because of the supporting cells get killed. And that's why we see often people recover their sense of smell and then lose it a couple of weeks later and develop parosmia thereafter. So that's the nerve destruction. But if the people who lose their sense of smell, they get nerve destruction, but they don't have this disordered regeneration that we think is happening with parosmia, then they're going to just have a normal, just regenerate their smell slowly and hopefully helped by smell training.
Mm. I mean, it has to be said that that thrown into the mix with all of this is what we've seen over and over in the Facebook groups, is that people experience these fluctuations, which are very distressing. As far as I'm aware, we don't yet know exactly what the mechanism is behind that.
No.
And so in addition to people transitioning from, typically the pattern would be total smell loss, then a certain amount of recovery where people report, oh, I'm getting better. And sometimes they'll say, oh, I'm almost completely recovered. And we all know, it has to be said, that the way people self-report their sense of smell is not always the way an objective smell test would rank them. But so you have this, so you transition to something where it's all looking good and you're smelling pretty well, and then suddenly you develop parosmia, and then boom, you get a fluctuation which takes you down to 0 again. then that's distressing. And then as it comes back, is it still, do you have parosmia? Are things beginning to smell more normal again? And this seems to be going all over the shop. So I think that that, you know, we have to consider that as well. Extremely distressing for people that are experiencing it. And of course, every time you go into a dip, you think, oh, this is it. That's the end of my sense of smell. And hopefully that's not the case. There are plenty of people out there who have been through a whole series of fluctuations, sometimes lasting weeks, and have still come out the other side. So that's the good news. But yeah.
We've had a few questions here on memory. And if you would, will the memory of correct smells fade away and forget? Because what you need to remember is with parosmia, you're not experiencing how coffee would smell. And I can really feel for someone that doesn't get to smell coffee in that way. So what's your thoughts on that? Perhaps, Simon, will that ever come back, that memory?
I think you should ask the other experts. I mean, I think for some people, okay, I'll stick to my guns. You should ask some of the other people here.
Speaking for myself, so as you probably all know by now, I have lost my sense of smell twice. Once in 2012, and then again with COVID last April. So I had a long recovery from the first time around. So 2 years to get an even functional sense of smell, and then another 6 to really get myself out of the worst of the parosmia and feel like I had a good quality of life. And here's an interesting thing that I have found. So I was interested in smell training very early on. Smell training is now part of my daily routine, and I'm very in tune with my sense of smell. I think I score as normal with the sniff and sticks test, but I still have a level of parosmia, and I've had increased parosmia as a result of COVID-19. But what I do think that smell training gave me was the ability to be much more elastic in the way I could conjure up a smell. And I think that that is a talent that, you know, it's a thing that you can learn to do. And it starts with smell training. So smell training is something that you do with your mind as much as do with your nose. And I now find that I'm able to conjure up all kinds of smells. I mean, even from my remote past, things that you think, I mean, that I haven't really smelled that many times in my life, but I do think that it is a, you know, something that you can learn. And I would just, to the person who asked whether they would forget what things smelled like, I would say, smell training will help you with that. Right, another question.
Yep. Going back to smell training, what about smell training with things that are parosmic to help speed up the activation of the damaged cells? Now, I think we've had something similar before, and that's an interesting one.
Yeah, well, we don't really know that, but I mean, you know, I've really struggled with coffee, and then I thought, no, I'm just gonna, I have to drink coffee, so I'm just gonna have to find my way back in with coffee.
I guess,
and I just did. I think there are a lot of people in the Facebook group who would say, just push your way through and keep trying. And the first mouthful is really awful, and then by the second or third mouthful, you've somehow gotten over that hurdle. And
yeah, I guess it's quite hard to ethically try and study something like this, because this is parosmic, it's discussed. It's actually quite hard to study that on people. So yeah, it's a hard one to say.
It's an interesting one. It might go two ways. You know, it might help you in that you might get used to that sense of disgust, and you might get start to get a better, start to moderate the horrible smells you're getting from coffee. But on the other hand, if you think of what we think is going on with smell training, you're encouraging neurons to grow, you might end up growing more of the coffee ones, and they might be getting it wrong. So you might end up worse off. Now, we don't think that's the case. And hopefully as you grow more of them, they kind of find the right pathway. but it could go either way. So it's a big question, and we'd love to try it out.
I mean, certainly I have to say, for my own part, I'm N equals 1, so not really very reliable. But you know, eventually, coffee smells okay to me, and my brain is kind of filled in the blanks, and it's not an unpleasant experience. And when I open the jar of coffee, when I make the coffee in the morning, it smells nice and like it's breakfast time, and it's okay. And I do think that, for me anyway, I have habituated to all these things, and maybe they're a little bit different, but the disgust is gone. And I think that maybe we should think in terms of finding a way to switch off the disgust rather than just talking about these individual molecules that really may be the looking like the culprits at the moment.
I mean, I don't think I've heard of anybody that's told us they've got worse if they've persisted with the coffee.
Yeah, I would agree.
On balance, the people that persist with the coffee are the ones that get on better.
And can I just say a great tip that was offered by somebody in the Facebook group, to drink, if you really feel that you need your coffee in the morning and you really can't bear the smell, to drink it through a straw. And that takes it into your mouth and down your gullet without kind of plaguing your, you know, your sense of smell too much. So you might try that one.
We did
Inject the caffeine directly.
Yes, right.
Yeah, we did a really interesting poll where we asked people which was worse, instant coffee or fresh, and that came back as fresh most of the time, but sometimes instant. And I think that depends on which molecules are triggering. And the other part of that poll was, does it smell, which is worse, the smell or the taste? And most people came back and said smell. A lot of people said both the same, but most people said smell. But there were some people that said, no, the smells fine, but it's a taste that's horrible. And that's quite hard to explain. But in general, the smell, and I think the smell of the grounds is even worse. I think it's the people that can pick it up when they're walking past a coffee shop where they've been grinding the beans.
Yeah.
I think that's even worse because that is such an intense aroma.
It's exactly the opposite way around to the usual hedonics of coffee. Grounds smell fantastic. Fresh coffee smells, you know, brewed coffee, and then the taste is not nearly as exciting.
Yeah.
Well, for me anyway.
I guess it depends how fresh your coffee. If it's stale, then you'll lose a lot of those things. That's true. Jane, just we're obviously quite conscious of time, but Jane, is it common to have parosmia that's not disgusting, i.e. the distortions is bearable? Yeah,
we've had people describe that as well. They've said they've had aromas coming off the GCO and they've said, yeah, that's Prosmic, but it's not disgusting. So yes, it is possible. And yeah, I mean, it just gets, the more you look, the more difficult it gets because there's so many things we're finding out, and the more interesting it gets. I mean, it becomes even more fascinating as to why we've got all different modifications of it, really.
That's just so bizarre to me that you distinguish between Parosmic and, because obviously the smells will be altered, right? So you have essentially three things. You have things that smell, or four things: things that smell normal, things that smell, three things altered, but it's four things: things that don't smell normal, things that smell parosmic but not disgusting, and then things that smell parosmic and disgusting. But if you say that parosmia is disgust, then those two things should be lumped together. So they have the percept of the same smell, because often they will report the same smell. And again, Chrissi can chime in on this, for their parosmia smell. That's very interesting. And yeah, like you say, it just makes it even more complicated.
Yeah, there's a lot of stats involved for all these kind of things, isn't there?
I think the sense of disgust is way above, you know, normal mix can smell something and say it's disgusting, but the sense of disgust that you get from Prosmic is much more of a reaction. It's a different magnitude, it's on a different level.
Yeah. I'm afraid we're getting to the end of the hour.
Yeah, so many questions this time.
So many questions. We haven't even scratched the surface. There will be more of these webinars. Do please, come back the next time. We may be able to continue this discussion in 2 weeks. We'll see. We've got enough material, I think, to carry on for many weeks after that. So I would just like to say, for anyone that arrived late and didn't hear my announcement at the beginning of the hour, we have a new platform at AbScent, and you can find it if you scroll all the way up to the top of the chat bar. You'll find a link. Joining the AbScent network is free. We've got a lot of good stuff going on there, including some master classes. So do please check that out. Otherwise, please join our Facebook groups, where you will find us easily under the name AbScent. So we've got 4 groups there. So I would just like to say again, thank you to my guests. Thank you, Aidan and Jane and Simon, for making this such a lively information exchange. And we hope to see you another time soon. So that will be it. Good night, everyone.
Thank you.
Good night,
Bye-bye.
everyone.